Hello, everyone. It's Mr. Vallejo. Welcome to science class. Today, we'll be doing the macromolecules test laboratory activity. And so let's start with some background information, and then I will do the test with you. All right, here we go. Let's go and share screen and go to this pre-lab discussion for the basic information. All right. There are 4 different types of organic molecules. We'll be testing for 3 of those today. We'll be taking a look at carbohydrates, and we're going to test for 2 different types of carbohydrates. And we'll test for lipids. We'll run a test for proteins. The 4th type of organic molecule is the nucleic acid, like DNA or RNA. Since we'll be doing so many experiments with that, we will not cover that in this particular macromolecules laboratory. All right. So with carbohydrates, you have monosaccharides, disaccharides, and polysaccharides. We are going to take a look at the monosaccharides and polysaccharides specifically today. So let's take a look at the monosaccharides. An example of monosaccharide, a monosaccharide is glucose. Glucose is also called dextrose. If you take a look at a picture, there's some honeybees, and they are in their you can see the honeycombs there in their hive, and you can see that there's honey in there in the combs there. The honey is made of glucose. It's a very high energy source. It's a basic food molecule, not just for bees, but for people as well. Also, monosaccharides make up all carbohydrates. So mono is a prefix that means one. Saccharide means sugar unit. So monosaccharide has 1 sugar unit. A disaccharide has 2 sugar units. So a monosaccharide is sometimes called a simple sugar, and a disaccharide, is called a double sugar. There's some other ones like an oligosaccharide you might see when you take a look at your Gatorade bottle. Sometimes they put some oligosaccharides in there, so there's just some short chain saccharides, short chain sugars. But we're going to test also for the long chain sugars. Those are the polysaccharides. Polysaccharides are made up of just multiple short polymers and even just linked up simple sugars. So to make a polysaccharide, which is a long chain, so it's a polymer, you have a bunch of monosaccharides that are stuck together, and they're stuck together by a chemical reaction called the dehydration synthesis, and you'll see that right here. It's called the dehydration synthesis because water comes out of this reaction when the hydrogen and the hydroxyl group come together. You get that water coming out. Some people call it condensation reaction as well. So it's a polysaccharide. We'll test in this lab for monosaccharides and polysaccharides. But note that the polysaccharides are made up of just a long chain of monosaccharides. The starch stores energy in the form of sugar for later use in plants. Lots of different foods we like as people are starchy. You can think of starchy foods like rice or pasta, or bread. These are all starchy foods, and they have polysaccharides. So the job of the starchy foods, the job of the polysaccharide, to store energy, and also starchy foods are used as structure in plants. The 3rd group is the lipids, and we're going to test for the presence of a lipid today. Lipids, their main job is energy storage. You can see they're very effective at storing energy, but they also can cushion organs and also are for support. but their main job is for energy storage. They come in different forms, come in fats and oils. Fats are solid room temperature and typically come from animals like this lard. And the oils are typically coming from plants and they're liquid at room temperature. An example is EVOO, extra virgin olive oil. An example of an oil. All righty. And proteins are essential to the structures and activities of life. Your body is 70% water, but then after that, it's mostly protein. Proteins are polymers, so they are also long chains, but they are made up of not monosaccharides, but they are made up of amino acids. So today we're going to do tests we're going to do 4 different tests, one for the monosaccharide, one for the simple sugars. And what you're going to do is you're going to start out with the solution. And it looks like this blue one here. And after heating, if you see a color change to green, orange, or red, you'll know that you are in the presence of a monosaccharide. So we're looking for a color change today, green, orange, red, to give you a positive indication, indicator that you have monosaccharide in your solution. For polysaccharides, we're going to run the Lugel's iodine test. What you're going to see is that it's going to turn a dark brown or black. Take a look at the first test tube here. You can see that this is what the iodine looks like. This is the Lugol solution. This right here, the second test tube, has starch in it. When you combine the two, this is what you get. So this is the color of a positive indication for the presence of starch. For lipids, instead of doing a chemical test, we're going to put it on a piece of brown paper. And if you get a translucent grease spot after the liquids dry off, that is an indication of the presence of a lipid. And then finally, the test for the proteins will give you a pink or lavender color. You can see in the picture, you can see that you have a dark blue test called Biuret in the presence of proteins. These guys turn purple. And this is a quantitative test, and it'll turn to richer, darker shades of purple in the presence of more and more proteins. Okay. All right. So that is what we are going to do today. So if you have the handout that looks like this right here, let's switch cameras and so you can see it a little bit better. And view this. There's the background information that you have on the different chemicals that we've just gone over: carbohydrates, lipids, and proteins. You also have the opportunity to make a hypothesis, because what we're going to do is we're going to test water, oil, milk, oatmeal, juice, and today it's orange juice instead of apple juice. But we're going to test those 5 foods and then also test for an unknown a little bit later. So why don't we pause for a second and go ahead and put a check mark here if you think that water contains lipids, carbohydrates, or proteins, and do that for the other 4 solutions as well. So I'm going to pause here and then we will come back after you made your hypothesis. All right, so let's go ahead and do the test. Here's all the procedures and the handout that you can see here. Also, we need you to complete this table. There's a table here for you to record whether on the first test, whether it leaves a spot or not. And then on the other test, color right here. And then from the pre-lab, if it turns a correct color, we can say whether it has Benedict's or monosaccharides, whether it has a polysaccharide, whether it has a protein. And over here, if it leaves a spot, you can put a plus if there is a lipid present, a fat or an oil. So let's go ahead and take a look at the first test. So what we're going to do is we're going to take this brown piece of paper, and on this brown piece of paper, we're going to divide it into 6 squares: water, oil, milk, oatmeal, juice, and unknown. Okay, I'm going to let that sit until the end of our time together. So I'm going to move this out of the way and we'll come back to this after we spot it. Let's go ahead and spot it. Let's go with a drop of water. Let's see. Here's a dropper bottle with water in it. Okay, I'm gonna put a drop right there. All right, a drop of vegetable oil. Here we go, right here. All right, I actually got some down there with the juice. All right, we have milk over here. Drop of milk, drop of an oatmeal solution. And we're going to try not to get the actual oatmeal, but the liquid that is in this bottle. Oops, 2 drops or 1 big drop, we're going to call it. Okay. All right. The juice, as I said, is orange juice today. So let's take a drop of orange juice and put it right there. Alrighty. And then a drop of the unknown. What is the unknown? Well, it's, we don't know, but well, actually you don't know. I know because I made it. So that is the unknown X solution. Take that and drop. Right there. There's the unknown X. All right, so I'm going to set this aside. We'll let this dry, and I will show you that a little bit later. All righty. Our next test will be the on a spot plate. So let me get a paper towel out and put that on there. And then it's called a well plate or spot plate. What we're going to do is I'm going to add the solutions to this. Let's see, I have a pen handy to mark these. This will be water, oil, milk, oatmeal, and then I'm going to have juice and unknown X. Juice over here, and then the unknown X going to be right there. Okay, that's for the first test over here. And then I'm going to run the other test here with water, same chemicals, water, oil, milk, oatmeal, juice, and then unknown X. So there we go, all labeled so I know what's what. Ooh, all right, so we're going to put in the solution or in the well plates. But just a dropper full of each liquid will give you a good idea. Go a little further out. Ready? So here's a squirt full of milk. And then milk also over here. All right, I'm done with the milk. Do the same with the juice. There's the juice. It's half full juice. There's my juice down here. There we go. 2 out of 6. All right, for the oil, it gets a little icky, so instead of adding so much oil, I'm gonna put 5 drops oil in there. It will test just fine. 5, and then where's the oil over here? Here's the second one. 1, 2, 3, 4, 5. And then water, you want to add just enough water so that you have about half full. So, ooh, that's a little much. It's this one right here. All right, did better on that one. So now I've got water, oil, milk, juice. I need my oatmeal solution and my unknowns. So get that oatmeal bottle. Here it is, oatmeal. Take that, put the oatmeal in there, and mix that up a little bit. The oatmeal in there. Okay. So let's run the tests. We have 2 different tests. The first one is Lugel's iodine test. And 3 drops of Lugel's in each one. Here we go. 1, 2, 3. Alrighty. Oil. 1, 2, 3, 4. This is qualitative, so it doesn't matter that I have an extra drop in there. 2, 3. There's the milk. There's the oatmeal. 1, 2, 3. There's juice. 1, 2, 3. And then the unknown. You know, I didn't add the unknown, did I? So let's go back. I'm going to add the unknown to it. Let's see what color it turns color or not. There's the unknown there. And there's the unknown there. Okay. How about that? All right, so it looks like, let's get a little bit closer so you can see water and oil don't look like they have changed to the dark brown black color. Look at that, the bottom of the oil though, it seems like a chunk. That's not it there. This one over here, the oatmeal definitely has starch in it. Milk? No, I don't think so. But the unknown X, I think does. And the juice, is juice starchy? Well, juice has sugar in it for sure. But as far as starch, you know, the juice has a simple sugars, the monosaccharides in it, but not any starch. That looks like about the same as milk. You don't usually think of milk as a starchy substance. These two are about the same color. But the unknown X is a little bit darker than these guys, so I'm going to call that a positive. And that oatmeal, I'm going to call that a positive as well. And focus it a little bit better. Yeah, it's better. Okay. And the juice from the lighting looks like it's a little darker, but it's not really. I just part of the experiment that if you were with me, you could actually see it. Let me see. It's still not. This is a lot noticeably lighter than this. Okay, so let's test the other one. The other one is a protein test. I'm going to add some bio reagent to to each of these 6 wells. First, the water. 1, 2, 3, 4, 5. Remember, we're looking for a purple color. There's oil. 1, 2, 3, 4, 5. Okay, pretty blue so far. Is there protein in milk? So check that out. 1, 2, 3, 4, 5. Oatmeal. 1, 2, 3, 4, 5. Got the same color as those two right there. Here's the juice. 1, 2, 3, 4, 5. And then here's the unknown. 1, 2, 3, 4, 5. Ooh, look at that. Okay, so let me see if I can get some of the shine, the glossiness off of our picture. Sometimes this works better when you have a white background. So I'm going to slip this under. You can see that the water and the oil, definitely not. Milk is purple. This is milk, oat, juice, and unknown. So having done that, you can see that the milk and the unknown both have a positive test for the Biuret test. Remember, Biuret test for the presence of a protein. All right, so good deal on that. Let's do our final test. And for a final test, we need these test tubes. So there you go. I'm going to pause for a moment because I need some hot water. All righty, guys, I'm back and I have my hot water right here. I just put the water in the microwave for 2 minutes and it's not boiling, but it is hot. You might see a little bit of steam coming off of that. So that is the hot water. What I'm doing on the Benedict's test is going to add the different chemicals to the test tubes. And then I am going to add 5 drops of the Benedict solution. So I have over here the juice. Let's see, I'll just take these out and put them in the beaker as I do all that. So here's the juice right here. So there's that one. Take these out. Here's some juice. This will be the oatmeal solution. I'm going to take that and mix that up again. Since it's been sitting a little bit. I'll make sure I want to get the objects that I'm trying to test in there. So here's the oatmeal. Got an oatmeal chunk that I don't really want. I just want the liquid. It's a little difficult to do since I shook it up. So here's the oatmeal. And there we go. Oatmeal in there. This one is water. So I'm going to take my water and add some water to this test tube. So here's the water right here. And if I wanted to be precise, I could count 20 drops, which would equal 1 milliliter. But there's enough water right there. This is the unknown. So let's check the unknown. Let's put that right in there. All right. Fourth one, to the left. Oil is since this is qualitative, it doesn't matter that I'm not adding exactly the same amount. It gets a little icky with the oil, so I'm just going to put 5 drops in here. 1, 2, 3, 4, 5. That'll still give us enough to give us an indication whether we have the Benedict solution, the monosaccharide from the Benedict solution test. And here's the milk. So I'm going to take a squirt full of milk, put it in there. So here are my 6 test tubes right here. I'm going to add 5 drops of the Benedict solution. Remember, we're looking to see if it might turn orange or yellow or red, even green. So let's take the cap off of the Benedict solution, add 5 drops to each. 1, 2, 3, 4, 5. 2, 3, 4, 5. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. And 1, 2, 3, 4, 5. Good. All right, so now we have the Benedict solution in all 6 test tubes. Let's see. And there's number 1. You can see the color right there. Here's number 2, the oil. Okay. This is so that you can get the initial color. Here is the 3rd one is the milk. So here's the milk with 5 drops of the blue Benedict solution. All right, the 4th one is oatmeal. There's the oatmeal right there. 5th one is your juice. Okay, mixing that up a little bit. And the last one is the unknown solution, the unknown X. These are the initial colors for all 6 tubes here. Okay, and let's go ahead and put them in the hot water bath all at the same time. It only takes about 3 minutes to do. So let's go. Ready, set, go. That's going to pour off some of the water so these don't float. There we go. These guys are sitting right here. I don't really need this one anymore. These guys are sitting in hot water. All right, so 3 minutes, we'll be able to get our results while we're doing that. Let's go ahead and take a look at the lipid tests. We take a look at this, you can see that the drops have not dried completely yet. So let's go to another one that I did earlier, and you can see that definitely the oil had a translucent grease spot, as well as the X, water, milk, oatmeal, juice. They do not have a translucent spot. It completely dried out. And this X right here, that means there's going to be, in my particular mix of X today, you have the oil can be found in as part of the unknown X. All right. Alrighty. Let me take a look at the Benedict's test. Let's see how that's going. Remember, this test for the presence of a reducing sugar or monosaccharide. There's water. Water continues to be blue. There's the oil. It's also blue. Right there. It's 1 and 2. There's number 3. Already you can see something happening. Milk. But is it orange or yellow or red? It's a little purplish, isn't it? And number 4, still quite blue. That's the oatmeal. Okay, here's number 5, the juice. Guess what? Does juice have sugar in it? Does juice have a glucose in it, a monosaccharide? And already I can say, whoa, it's definitely a different color. And what about the unknown? Did the unknown have a yellow, orange, or a red? In this case, no. Okay. So we'll let that run a little bit. But that is basically our lab. Let's take a look at the data again real quick, and you can pause it, recording if you need to. Here is the first test with the lipids. You can see that there is a translucent grease spot at oil and at X. Here are the spot plate results. And with the spot plate results, I want to remind you that the First four plus these two right here were from the Benedict's or the iodine test, the Lugol's test for the presence of polysaccharides. And it looks like the positive one is this one right here. And this one right here. You can see that 3 and 5 look very similar, kind of coffee colored, but then give you the positive test like you see right here with the oatmeal and right here with the starch. Down here with the, we're looking for the purple color for the Biuret test. And here's water and oil, negative for proteins. But over here you have milk. Milk has protein in it. You have juice and, no, this is oatmeal and juice. Neither of these tested positive for proteins, but the unknown did. As you can see, it's a nice purple color right there. Okay. And then going back one more time to the Benedict's test, what you have over here is, let's see, here's number 2 and 3. Here's number 1. You see 1, 2, and 3. You do not see a yellow, orange, or red color. Okay, there. And then for 4, 5, 6, here's 4. Looking for a simple sugar. Doesn't look like there's a simple sugar in there, but there's definitely one in the juice. You can see it is very apparently different from all 5 of the other tubes. Alrighty. So monosaccharides, we did 4 different tests for the monosaccharides, and I hope you learned something today. I'm Mr. Vallejo, and thank you for coming to science class today. Remember to fill out your worksheet, enter those into your learning management system, whether that's Schoology or Canvas. All right. Have a great day. Bye now.